Hello, this is the Gel Electrophoresis Lab. Our lab group investigated the separation of proteins through the method of gel electrophoresis. To begin the process of protein separation, a voltage is applied to the gel containing protein samples, forcing migration of the protein through the gel. It is noted that shorter molecules move faster and further through the gel than larger molecules. The protein samples are denatured using beta-mercaptoethanol, also known by the acronym BME, which allows the disulfide bonds within the proteins to be cleaved and subunits separated. This ensures the movement of the proteins through the gel is only dependent on size. Some field applications of gel electrophoresis include DNA analysis within forensic labs or protein identification.
Here's a nice metaphor for gel electrophoresis. This open field is like water, or some sort of buffer, and you have proteins, the rabbit and the deer. They can move freely until something like barbed wire comes. This barbed wire causes them to stay put. They would only move unless there was a strong force. There's a wolf. This is like an electric voltage. The deer can only make it so far. The rabbit makes it much farther. This is like migrating proteins of different sizes.
The first step in this process is to hand cast the polyacrylamide gels. First, add Resolver A into the mixing test tube. Immediately after, add Resolver B into the same test tube and mix thoroughly. In a new test tube, add stacker A and B to form the stacker solution. Clean the glass plates with ethanol and a Kim wipe. Then construct the glass cassette apparatus. Ensure the bottom of the glass plates are level with the bottom of the frame to prevent leakage. Place both combs in the top slits of the glass cassette apparatus. Next, mark 1 cm below the comb with a marker. This will be the level at which the Resolver gel is filled to. Next, add the APS to the Resolver gel solution previously made. Now, add the Temid to the same solution. Mix well. Next, carefully pipette the solution into the cassette apparatus. Fill to the line previously marked. After filling the cassette with Resolver solution, overlay the solution with IPA. Allow Resolver gel to polymerize.
Next, add the APS and Temid to the stacking gel solution. Mix well. After draining the IPA from the top of the cassette, pour the stacker solution on top of the resolving gel. Insert the comb into the cell once the stacker solution is added. After waiting until the gel is fully formed, remove the comb, exposing the empty wells. To ensure maximum protection, wrap all bottles with parafilm after use. As the gel is being prepared, begin protein sample preparation. First, perform the necessary dilutions for the unknown protein samples. Add the necessary amounts of Langley sample buffer and beta-mercaptoethanol and label each tube. Heat the protein samples in a dry bath for 5 minutes to finish the denaturing process. Prepare the running buffer using 100 ml of buffer solution and 900 ml of DI water. Assemble the electrophoresis chamber, then pour the buffer solution into the tank and chamber. Be sure to label each well. Utilize the 2 outside lanes for the standard solution. Pipette each protein sample into its respective well between the standards. After assembly is complete, connect the cell to the power source and set to 300 volts. Due to the high voltage running through the system, be wary of electric shock. Be sure to allow protein samples to migrate to the bottom of the gel before conducting the imaging process.
Once the protein samples have migrated to the bottom of the gel, the power source may be shut off. Carefully remove the top of the chamber, allowing access to the frame containing the gel samples. Dump the remaining buffer solution into the chamber. Disassemble the frame to allow access to the glass plates holding the gels. Using the scraper and DI water, carefully separate the glass plates and move the gel onto the imaging plate. Insert the gel sample into the imaging machine. Lastly, run the imaging software to obtain photos of the various sample migrations across the gel.
The first step in data analysis was determining the molecular weights of the unknowns. Here is our image gel, and as you can see, there are two standard ladders on the outside of the gel. These surround the three unknowns, which were U, J, and N. N actually had a known concentration of 20 nanograms per microliter. Using the known molecular weights in kilodaltons of the standard ladder, we were able to deduce what the molecular weights were in these unknowns. This was done using a semi-log linear plot. This was repeated for 4 samples, and the average was taken along with a standard deviation. This is plotted here below. Using this plot, we then tried to identify the unknowns from their molecular weights. It appeared that sample U might be bovine serum albumin. This is a protein derived from cows and is used as a concentration standard and a nutrient for microbial culture. It has a molecular weight of 66 kilodalton, which would give us 15% error. The other samples were thought to be the same collection of proteins. This is casein. Casein is actually made up of 4 different subunits: alpha S1, alpha S2, beta, and kappa. The phosphoproteins range from sizes in 19 to 25 kilodaltons. It's used as a protein supplement, actually.
Thanks for watching. Teamwork makes the dream work.